Hi, my name is Casey Calkins, your outgoing chair of the APSA Program Committee. It is my pleasure to welcome you to today's APSAsode, where we will begin by announcing the award winners for the scientific portion of the APSA meeting. This will be followed by a message from Dr. Mary Fallot, the president of the APSA Foundation. As you know, the APSA Foundation is the philanthropic partner of APSA in support of pediatric surgical innovation and discovery, surgeon scientist career development, member education, and other efforts which ensures access, quality, and equity in the surgical care and lives of all children. It is a source of essential funds to promote education and biomedical research in pediatric surgery. It has truly served as a gateway for young scientific investigators to become competitive for major external grants, and Dr. Fallot will introduce three wonderfully talented pediatric surgeons who have received foundation funding and will thereafter sequentially review the results of their supported research. As you all know, the APSA Board of Governors made the difficult and necessary decision to hold this past year's 52nd annual meeting in a virtual format, which had been originally scheduled to take place in Phoenix, Arizona, with our president, Dr. John Waldhausen, presiding over the proceedings. Nevertheless, the scientific content was again outstanding and was presented during the 3 days of the main meeting, as well as throughout the subsequent APSAsodes, which I hope you have found enlightening and educational. Today marks the culmination of the scientific content with our three APSA Foundation scholars. For the 52nd annual meeting, the program committee received 329 abstracts for consideration, of which 103 were accepted to be presented in an oral format and 30 in a poster format. The latter were afforded a 2-minute oral summary of their work. This material remains available for all who to subscribe to the annual meeting in an on-demand format using the Whova platform. Ultimately, 40% of the submitted abstracts were accepted for presentation, which continues to our objective to accept more valuable content at the APSA meeting. In addition, this year, we added a medical student-only session where 10 medical students presented their work in a short oral format. The moderators of this session included Drs. Jill Whitehouse, Anthony Stallion, Richard Glick, and Christian Walters, who helped to select the award recipient. They heard 10 outstanding presentations during the APSAsode that aired on July 28th. And if you didn't catch it, I would encourage you to go watch some fantastic future pediatric surgeons present their work. The award winner for Best Medical Student Presentation during that session is Jennifer Crook of the University of Tennessee Health Science Center in Memphis, Tennessee, for her presentation entitled, "Why Do Ports Get Stuck? A Case Control Study." Congratulations to future Dr. Crook, who will receive a monetary award in the amount of $250. The next award is that of the Poster of Distinction Award, which this year is presented to one outstanding poster, whereby the recipient must be a trainee currently enrolled in a surgical training program, either a residency or a fellowship. Presentations were judged on scientific merit and visual presentation by members of the program committee. This year's recipient receives a prize in the amount of $250, and that recipient is Dr. Kyle Thompson from Boston Children's Hospital for his presentation entitled "Predictors of Anti-Reflux Procedure Failure in Esophageal Atresia Patients." His poster aired during the scientific session 1 on neonatal surgery, again available on Whova, and I would encourage you to go back and watch it if you missed it. The M. Judah Folkman Memorial Awards were created in honor of Dr. M. Judah Folkman and his legacy of leadership in our organization, and the tremendous body of work he left behind, and the way he selflessly shared his art of medicine with so many students, and the art of healing with so many children and families. The American Pediatric Surgical Association and the Association of Pediatric Surgery Training Program Directors selects eligible recipients who also must be trainees currently enrolled in a surgical training program, either residency or fellowship. 2 winners are selected for best oral presentations, 1 in basic science and 1 in the clinical categories. Winners receive a prize in the amount of $1,000 and a Folkman medallion. Again, presentations are judged on scientific merit and actual presentations. And here to present those awards is our immediate past president of the Association of Pediatric Surgical Training Program Directors, Dr. Ben Nwome. Dr. Nwome.
My name is Ben Nwome from Nationwide Children's Hospital. On behalf of APSA and the Association of Pediatric Surgery Training Program Directors, I am pleased to present the 2021 Folkman Award. This award was created in honor of Dr. M. Judah Folkman and his legacy of leadership in our organization, his tremendous body of work, and his selflessness in sharing the art of medicine and scientific discovery with his patients, his trainees, and his colleagues. APSA and the Association of Pediatric Surgery Training Program Directors are proud to present the Judah Folkman Memorial Award for outstanding research presentations that were given by general surgery residents or fellows during the just concluded 2021 annual meeting of our organization. We selected 2 winners, 1 in clinical and 1 in the basic science categories. These winners will receive a prize in the amount of $1,000 and a Folkman medallion. These winners are as follows. The clinical award goes to the paper Implementing a Standardized Gastroschisis Protocol Significantly Increases Primary Bedside Sutureless Closures Without Compromising Closure Success or Early Clinical Outcomes, by Maeve O'Neill Trudeau from University of Montreal, Saint Justine. The Basic Science Award goes to the paper In Utero Base Editing Ameliorates Pathology in a Mouse Model of Mucopolysaccharidosis Type 1 by Saurav Bose from the Children's Hospital of Philadelphia. Congratulations, and on behalf of APSA, thank you all for participating in our annual meeting.
Thank you, Dr. Nwome. Next, it is my pleasure to introduce Dr. Derek Wakeman from the APSA Surgical Quality and Safety Committee, who will present the APSA Quality, Safety, and Value in Surgery Award. This award is presented to the primary author of the manuscript that best demonstrates quality improvement principles, patient safety initiatives, and/or addresses the value proposition by demonstrating an improvement in outcomes, while at the same time reducing cost or other measures of resource utilization as selected by members of the Quality and Safety Committee. The recipient of this award receives a $500 prize. Here to announce that award winner once again is Dr. Wakeman.
Hello, my name is Derek Wakeman. On behalf of the APSA Quality and Safety Committee, chaired by Dr. Mehul Raval and co-chaired by Dr. Monica Lopez, I'm pleased to announce the winner and runner-up of the annual APSA Value in Surgery Award. During the May meeting, we announced the 2 finalists. As a reminder, these 2 projects were chosen from a pool of approximately 100 abstracts that focused on improving quality, safety, and value in children's surgery. The top 8 abstracts were asked to submit a full manuscript to be considered for the Value Award. The top 2 manuscripts were chosen as our finalists as they best demonstrate quality improvement projects which improved patient outcomes, reduced harm in healthcare resource utilization, and improved overall value. So the runner-up is Dr. Syed Arshad from McGovern Medical School at the University of Texas at Houston with a manuscript titled A Cost-Effective and Safe Quality Improvement Initiative for Pediatric Intussusception. The group demonstrated safe discharge of children to home from the emergency department after successful reduction of intussusception, preventing admission, thus saving healthcare resources. So the winner of the 2021 APSA Value Award is Dr. David Grabsky from the University of Virginia with their manuscript titled Reduction of Postoperative Opioid Use in Neonates Following Congenital Diaphragmatic Hernia Repairs, a Quality Improvement Project. Their group significantly decreased opioid administration to neonates after congenital diaphragmatic hernia repair and found a concomitant decrease in ventilator days after CDH repair. I'd like to thank everyone who submitted manuscripts for the Value Award. And as a reminder, content for these quality improvement projects can be found on the APSA Quality and Safety Toolkit with helpful advice and tools to implement related QI projects at your hospitals. We are looking forward to reviewing abstracts for the 2022 Value Award in a couple months. Thank you.
Thank you, Dr. Wakeman. Finally, it is my pleasure to introduce Dr. Bethany Slater from the APSA New Technology Committee, who will present the Innovation Award, which is open to any presentation author, regardless of level of training, of employment, and judged on the uniqueness and potential impact of the idea, as well as the quality of the presentation. In other words, attending staff are eligible for this award. The winner is selected by the New Technology Committee and receives a prize of $500. Again, to introduce that award winner is Dr. Slater.
Hi, I'm Bethany Slater, and on behalf of the New Technology Committee, I am pleased to announce the Innovation Award. And the award goes to Dr. Oliver Munster from Munich for his abstract entitled The ABCs of Magnetic Esophageal Compression Anastomosis Using Specially Designed Curved Magnets for Minimally Invasive Repair of Esophageal Atresia. His presentation described 3 patients with esophageal atresia and reports the outcome of at least 1 year thereafter. If you were not able to view this presentation, please refer to the enduring material of the New Technology APSAsode. Congratulations again, Oliver. Very pleased with this well-deserved award.
Thank you, Dr. Slater, and congratulations to all of our 2021 award recipients. And now it is my pleasure to introduce Dr. Mary Fallot, your 47th president of APSA and current president of the APSA Foundation, who will offer some perspective on the APSA Foundation and introduce our APSA scholars. Thank you all for once again for your continued support of our organization and support of the annual meeting. Dr. Fallot.
Good evening, and it's great to have you with us. I have the fun of introducing the research portion of the program, where we get to hear about the great work that 3 of our recent APSA scholars are working on or have completed. We have 45 recipients of foundation awards to date. We've heard from several of these scientists, and their testimonials are on our website, and I would encourage you to look there to see the great work that's been done and the gratitude that's been expressed. But we also know that the return on investment for this group of investigators is at least $65 million in grant funding, which is an incredible track record for a small foundation like ours. We started a Twitter campaign to brag about our scholars, and it was recently launched in early October. If you aren't following us, we hope that you will, as we will feature one of our scholars every few weeks. Our first feature scholar was Dr. Besner. Gail was the first woman to receive a foundation grant, and the amount awarded was $7,500. She's now chief of pediatric surgery at Nationwide Children's Hospital and has 26 years of continuous NIH funding. You go, girl. But our ability to keep funding the scholars and also to fund other important initiatives that you've brought up depends on you. On Giving Tuesday, November 30, the Thursday after Thanksgiving, please consider joining our cafe club of sorts. We know most of you drink coffee and some indulge in caffeinated drinks that you disguise in a can, but all we're asking for is to consider donating $5 a week or about $250 a year to the foundation. This amounts to the cost of a fancy cup of coffee once a week. You can choose the payment interval, and all of these donated cups of coffee will go a long way toward fulfilling the mission and goals of the APSA Foundation. An important part of the mission and vision of the foundation is to support pediatric surgical innovation and discovery. If you're a newer member of APSA, please pay attention to my next message. The grant guidelines will be released in December for next year's scholars. Please look for the announcement in email and on our website, and we'll also announce it on Twitter. Our busy research committee does the judging with recommendations back to the foundation board. The current chair and vice chair of the research committee are Doctors Sean Kunisaki and Bo Lavorne. The preferred applicant is a regular APSA member, typically less than 5 years in practice or less than 40 years old. You cannot receive it twice. The application period is open about 6 to 8 weeks and starts in mid-December. The award is $25,000, and we hope again to award 3 scholars with announcement of the winners at the annual meeting. This year, we plan to have separate categories for basic and clinical or translational research. Earlier in the year, I announced that these 3 young men had won the scholarships for 2021, and next year, you'll hear about their projects. They are Dr. Augusto Zani from Toronto, Dr. Hao Liu from the University of Wisconsin-Madison, and Dr. Young Yoon from Stanford University. But now I'd like to briefly introduce the 3 young women who will present to you this year. The first is Allison Spear from McGovern Medical School at UT Health Science Center in Houston. She will speak about her project defining the role of the enteric nervous system and epithelial barrier function in human intestinal organoids. Dr. Krista Grant from Penn State Children's Hospital in Hershey will report on her project, Contactin-1, a potential biomarker and therapeutic target in high-risk neuroblastoma. And Dr. Emily Partridge of CHOP will report on her project, study of cerebral autoregulation and pumpless arteriovenous extracorporeal membrane oxygenation. So without further ado, let's hear from our scholars, and then please stay tuned for a live question and answer period at the end, moderated by Drs. Kunisaki and Lavorne. Put your questions in the chat, and we'll save them to the end. And thanks again for tuning in. Have a good evening.
Thank you to APSA for the opportunity to present our research. As we know, intestinal failure is a devastating disease with increasing incidence, and it's most often due to loss of bowel length. Common etiologies include necrotizing enterocolitis, gastroschisis, intestinal atresia, and malrotation with volvulus. We know that contemporary management includes parenteral nutrition, bowel lengthening surgery, teduglutide, as well as intestinal transplant. And these treatments in the institution of multidisciplinary care have increased survival, but morbidity and cost remain significant. There's a desperate need for more effective therapies for children like these. Tissue-engineered intestine is a promising solution. However, complex intestinal functions have not yet been confirmed in human intestinal organoid or HIO models, and this knowledge gap limits our ability to utilize this as curative therapy. Thus, understanding the processes required to generate a functional tissue-engineered intestine are a high priority. Our lab utilizes an HIO model, first described by Jason Spence, as well as my collaborator, Noah Schroyer. We're able to generate HIOs in vitro from human embryonic stem cells and transplant these underneath the kidney capsule and grow them in vivo in immunocompromised mice. We found that they grow from about 100 microns to 1,000 microns after a month in vivo. You can see an example of this in panel A. After 8 weeks in vivo, they grow to about 1 centimeter, which you can see in panel B. Our H&E staining demonstrates the profound maturation process that they undergo in vivo, demonstrating crypts and villi, as well as epithelial and mesenchymal development. And CDX2 marks the intestinal regional identity of both HIOs in vitro and in vivo. We know that intestinal functions are complex, including absorption, barrier function, and peristalsis. We're most interested in barrier function. Thus, we first utilized our model to evaluate tight junction genes. Here, we performed RT-qPCR on HIOs in vitro for 1 month, as well as THIOs in vivo for 4 and 8 weeks. We found that transplantation of HIOs promotes expression of these select tight junction genes, including claudin 3 and 15, occludin, and ENZO1. We then conducted immunohistochemistry on HIOs in vitro in the top row and transplanted HIOs in vivo in the bottom row. And we found that transplantation demonstrated more mature morphology and a paracellular distribution of these tight junctions. However, these HIOs and transplanted HIOs do not have an ENS, and we know that intestinal functions require the ENS, including barrier function. Thus, we became interested in incorporating an ENS into our system. We modified a protocol from Farnak Fatahi's lab, where we can grow enteric neural crest cells, or ENCCs. These mark for enteric neural crest cell marker SOX10, as seen in panel D, and they mark for vagal markers as well as enteric neural crest markers on PCR on the right. They also differentiate into enteric neurons as well as enteric glia, marked by TUJ1 and BOPB in the bottom left. And Mike Byerline, a collaborator at UT Health, is able to do whole cell current clamping and demonstrate action potentials in our ENCCs. Thus, we modified our protocol to generate HIOs, but to also incorporate ENCCs into this model, both in vitro and in vivo. We found that our HIOs in vitro in the top row and transplanted HIOs in vivo in the bottom row allow survival of our ENCCs, which are marked in red for TUJ1. And these incorporate into the HIOs into the mesenchyme just beneath the epithelium, which is marked in green for E-cadherin. Thus, although my long-term goal is to develop a functional tissue-engineered intestine for children with intestinal failure, my short-term goal is to understand the development and function of the ENS within this HIO model, and for this award specifically as it pertains to barrier function. Our central hypothesis is that the ENS is required for intestinal barrier function in HIOs. Aim 1 was to determine if the ENS is required for adequate production and appropriate morphology of tight junctions in the transplanted HIOs. And aim 2 was to define the effects of the ENS on intestinal epithelial permeability in transplanted HIOs. Regarding aim 1, we compared transplanted HIOs with and without neurons and looked at several select tight junctions and found that both claudin 3 and ENZO1 had upregulated expression in the presence of an ENS, whereas claudin 7 and 15 and occludin were unchanged. We then performed immunohistochemistry to further evaluate these same tight junction proteins. You can see transplanted HIOs without an ENS in the top row and with an ENS in the bottom row. And their expression looks fairly similar. However, I caution you in this because the heterogeneity of the tissue development makes it a little bit challenging. We then took THIOs to evaluate intestinal barrier function and placed them in an Ussing chamber. Our Ussing chamber then measured transepithelial resistance as well as short circuit current between transplanted HIOs with and without neurons, and we found no difference, which was a bit of a surprise. We then further evaluated permeability using FITC dextran. We placed this on the luminal side of our Ussing chamber and then collected from the serosal side every 15 minutes for an hour. And we found no difference between our THIOs with and without an ENS. And again, this was a bit of a surprise. In trying to understand why we did not find a difference in our THIOs with an ENS, I think this can best be explained by the variability in tissue development. You can see a THIO without neurons in panel A and with neurons in B, C, and D. These are 4 separate THIOs. Epithelium is marked in green with E-cadherin, mesenchyme in red with SMA, and neurons in white with TUJ. You can see that epithelial, mesenchymal, and neuronal development are all quite variable in all 4 samples. Thus, in conclusion, we know that THIOs with an ENS upregulate tight junctions claudin 3 and ENZO1. However, protein expression, morphology, and intestinal epithelial barrier function are much more challenging to evaluate in THIOs given the heterogeneity of our tissue. We plan to further evaluate ENS development with 3D advanced imaging to quantify glia and neurons. And then the ENS presence will be evaluated with GFP-labeled ENCs prior to our future Ussing chamber experiments to ensure less heterogeneity in our tissues compared. I'd like to again thank the APSA Foundation for their award, as it enabled the generation of preliminary data for my KO8 application submission, which is currently under NIH review. Thank you again to my lab, funding, collaborators, and scientific advisory committee.
Thank you for the opportunity to present our research, generously funded by the APSA Foundation. Neuroblastoma is a clinically and biologically heterogeneous childhood tumor with a range of clinical presentations and outcomes. It is the most common extracranial solid tumor affecting children, and the outcomes remain dismal for high-risk patients despite advanced multimodality therapies. Neuroblastomas arise from neural crest cells of the paraspinal ganglia and the adrenal medulla. Therefore, there is heterogeneity in the tumor presentation and location. In addition to genetic and biologic factors, such as amplification of the MECAN oncogene, tumor site is a strong independent predictor of survival in all neuroblastoma stages and risk groups. Adrenal tumors have the worst outcomes, while tumors arising from the thorax, neck, and pelvis carry a more favorable prognosis. The survival advantage of patients with extra-adrenal tumors compared to adrenal tumors is well documented in the literature, but few researchers have investigated the underlying mechanism. The overall goal of our research is to identify targetable factors associated with adrenal tumors and poor outcomes, as well as protective factors in known favorable sites such as the thorax. We approached this by first interrogating two large publicly available neuroblastoma data sets, namely GEO and Target. The Target data set is the only one that includes tumor location as a data point. We separated the 1,076 entries by primary tumor sites and then analyzed the outcomes shown here. Thoracic tumors and other extra-adrenal primary tumor sites were associated with improved overall survival, as well as event-free survival, compared to adrenal tumors, and this is in line with the previously published data. There were few numbers of the cervical and pelvic tumors, so those were excluded, and all further analyses were performed between the adrenal and the thoracic tumors. This heat map shown here demonstrates the differential gene expression between those two groups. From this, we aim to identify common factors that could account for the survival differential that we noticed between tumor sites. When analyzed for enrichment, the most common enriched pathways were related to cell adhesion molecules and included biological processes such as cell-cell adhesion, as well as cell junction assembly. 5 of the 8 genes overexpressed in adrenal tumors were associated with significantly lower overall survival, while 80 of the 145 genes overexpressed in those thoracic tumors were associated with significantly improved overall survival, again, driving home the point that thoracic tumors are more favorable. We chose to focus on Contactin-1 as it was one of the genes overexpressed in thoracic tumors and associated with improved outcomes overall. Additionally, Contactin-1 expression inversely correlated with MECAN expression in both data sets. What is Contactin-1? Contactin-1 is a neural cell surface adhesion protein present on normal neurons. The role of Contactin-1 in cancer is controversial, but its role in neuroblastoma remains unknown. In the Cancer Cell Line Encyclopedia shown here, Contactin-1 mRNA expression was highest in neuroblastoma among all tumors. Therefore, we hypothesize that Contactin-1 may function as a tumor suppressor in neuroblastoma by inhibiting cell migration and invasion. To study the effect of a tumor microenvironment, we developed a new model of extra-adrenal neuroblastoma, whereby neuroblastoma cells are injected subpleurally into the left posterior chest. The control group had neuroblastoma injected into the left adrenal gland as previously published. Tumors were explanted at 4 weeks. RNA sequencing on the resultant adrenal and thoracic tumors demonstrated that gene expression was significantly altered just by injecting the same neuroblastoma cell into 2 different locations. These findings recapitulated our initial findings in the human neuroblastoma dataset and demonstrated the relevance as well as the novelty of our model for future study of the microenvironment in neuroblastoma. After validating contactin-1 protein expression in 7 human and 3 mouse neuroblastoma cell lines by Western blot, we performed CRISPR-Cas9 mediated gene knockout of contactin-1 in 2 human neuroblastoma cell lines. 1 MECAN amplified cell line, SKNDZ, and 1 MECAN non-amplified cell line, SKNAS. Knockout was successful as validated by Western blot and did not affect cell viability under normal culture conditions. We induced adrenal tumors by implanting these 4 cell types into the left adrenal gland of NOD-SCID gamma mice. That is 4 total comparison group, 2 cell lines. Tumor growth was assessed weekly using IVUS, and the mice were sacrificed at week 4. Following necropsy, histology and other investigations were performed. Our results are shown here. First, a blinded pathologist graded the H&E slides of the 4 groups. There was no significant difference in gross tumor vascularity, tumor hemorrhage, or apoptosis between the groups. Again, our overarching hypothesis is that loss of contactin-1 is protective. So we expect, Again, our overarching hypothesis is that contactin-1 is protective, so we expect that contactin-1 knockout would promote tumor growth and metastasis in vivo. In tumors derived from the MECAN non-amplified cell line, SKNAS, our findings were in line with this hypothesis. There was no difference in tumor size, but there was a decreased survival in the contactin-1 knockout and a trend towards increased liver metastasis in the contactin-1 knockout. However, surprisingly, in tumors derived from the MECAN amplified cell line, SKNDZ, we found a significantly decreased tumor size in the contactin-1 knockout group, albeit with no significant difference in the survival or the liver metastasis. Of note, there was no liver metastasis in any tumor derived from the SKNDZ cell line. It is unknown if MECAN amplification would have an effect on the contactin-1 phenotype, but our human database findings suggest an inverse relationship between contactin-1 expression and survival in the MECAN amplified subgroup. So what is the underlying mechanism? To investigate this, we performed RNA sequencing on the tumors from both the parental and the contactin-1 knockout human cell lines, again, 4 groups. In both cell lines, there was downregulation of genes related to neurogenesis, synaptic development and function, and there was upregulation of cell adhesion and cell junction, as well as locomotion genes. The PI3-AKT pathway was a dominant upregulated pathway with a possible link to focal adhesion. In summary, our findings suggest that contactin-1 is protective in neuroblastoma, at least in the MECAN non-amplified tumors. The next steps in our project will attempt to elucidate the mechanisms by which contactin-1 regulates tumorigenesis in both thorax and adrenal tumors, with a focus on investigating the role of the PI3-AKT pathway. Ectopic overexpression of contactin-1 will be a first step in determining if manipulation of contactin-1 levels could be therapeutic. Thank you.
Good evening. My name is Dr. Emily Partridge, and it is my privilege to share my work, which was funded by the Grosfeld Award on study of cerebral perfusion in the lamb model of congenital diaphragmatic hernia. Despite improved mortality, long-term morbidity in patients with CDH remains high. Neurodevelopmental impairment affects 1 in 4 survivors of CDH. These children have significant impairment in motor function, cognition, hearing, language, social and emotional function, and a 9 times higher incidence of autism spectrum disorder than the general population. Identifiable prenatal predictors of neurodevelopmental impairment include fetal predictors of CDH severity, such as intrathoracic liver position, low observed to expected lung volumes, and low lung to head ratios. It has been hypothesized that in fetal CDH, changes in cardiopulmonary anatomy with resultant decreases in left ventricular cardiac output could cause abnormal brain perfusion, which has been shown on MCA Doppler studies in human fetuses. Whether this difference in cerebral perfusion impacts neurodevelopment has yet to be determined. Contrast-enhanced ultrasound is an emerging advanced imaging technology that quantifies tissue vascularity and microvascular tissue perfusion. It allows for precise perfusion measurements in specific anatomic locations not otherwise attainable by MCA Doppler. Measurable contrast-enhanced ultrasound perfusion metrics include the flux rate velocity, which is the rate of red blood cell entry into the cerebral vascular bed, transit time, which is the amount of time that a red blood cell spends within the vascular bed, and total global perfusion to a specific region of interest. The purpose of our study was to assess global and regional brain perfusion in the LAM model of CDH. Our focus began with in utero studies of perfusion in CDH lambs on placental support to establish baseline alterations in perfusion. We then turned our focus to studies of cerebral perfusion in lambs supported on ECMO. The LAM model of CDH is widely employed in our lab and includes the surgical creation of a diaphragm defect at mid-gestation or gestational age of 65 to 75 days in the lamb. After removing a portion of the diaphragm, the stomachs are delivered up into the thoracic space and the thoracotomy incision is closed. The lambs are then delivered near term and provide a robust model which recapitulates the pulmonary hypoplasia and hypertension so specific to this disorder. We created 5 CDH lambs at 70 to 75 days gestational age and used 5 healthy controls for our study. Near-term transuterine ultrasounds were performed under maternal anesthesia to measure umbilical artery and middle cerebral artery Dopplers. Fetal venous access was obtained by creating a small hysterotomy, shown by the large white arrow in figure A, and catheterizing a venous cotyledon leading to an umbilical vein. This was externalized as shown by the arrowhead, and the hysterotomy was then closed to provide for transuterine ultrasound. Ultrasound contrast agent was continuously infused, while flash replenishment acquisitions were obtained from a standardized view that included the thalamus and peripheral brain parenchyma, as shown in figure B, from which time intensity curve analyses were generated to extrapolate perfusion measurements. Standard fetal biometrics, including organ and body weights, were obtained at the conclusion of the study. There was no difference in umbilical artery pulsatility index between groups. Although there was no statistically significant difference in MCA-PI or calculated cerebroplacental ratios, they trended towards lower values in CDH fetuses. However, quantitative studies of cerebral perfusion by contrast-enhanced ultrasound demonstrated significant findings. CDH animals had significantly decreased global cerebral perfusion with increased transit time and decreased flux rate compared to control animals. Analysis of the central thalamic region, depicted in the middle row, shows markedly decreased perfusion with increased transit time and decreased flux rates. Measurements of peripheral parenchymal perfusion are shown in the lower row and demonstrate no significant changes in CDH compared to control animals. Biometric data confirmed significant pulmonary hypoplasia by lung weight and volumes normalized to body weight in CDH. Cardiac hypoplasia was seen in CDH animals, which is consistent with prior reports in the literature, and no differences in normalized brain weights were observed. Spearman's correlation analysis showed that global brain perfusion was most directly correlated with normalized lung weights. Due to the severity of the underlying pulmonary hypoplasia and pulmonary hypertension in CDH lambs, animals require support by ECMO to facilitate long-term study. This is analogous to the severest forms of CDH seen clinically. Support by conventional VA ECMO can be a life-saving modality, but is also associated with a number of complications, most of which can be attributed to the requirement for systemic anticoagulation. We have devised a simplified pumpless form of arteriovenous ECMO, which requires no systemic anticoagulation, with animals delivered by an exit to ECMO procedure and cerebral perfusion studies performed immediately before and after cannulation. Animals are monitored with continuous capture of hemodynamic status and regular blood gas and biochemical analysis. These studies have shown stable long-term support of the CDH lamb for up to 21 days with no thrombotic or hemorrhagic complications, with circuit flows, hemodynamics, and blood gas profiles similar to animals maintained on conventional support. We have recently completed cerebral perfusion studies on 5 CDH lambs cannulated onto pumpless AV ECMO compared to 5 lambs on conventional VA support. Complete quantitative global and regional analysis is ongoing at this time, but our preliminary findings demonstrate a dampening of global and regional cerebral perfusion after placement onto conventional VA ECMO, while no gross alteration in cerebral perfusion was seen in animals cannulated onto our pumpless arteriovenous circuit. Current studies are underway presently to assess the autoregulatory cerebral perfusion responsiveness to insults including raised intracranial pressure and induced hypercapnia and hypoxia in animals supported on pumpless compared to conventional ECMO. In summary, our studies of contrast-enhanced ultrasound showed a significant decrease in global brain perfusion and flux rate, with increased transit time in the CDH lamb in utero. The decreased flux rate indicates a lower velocity of blood entering the cerebral circulation relative to normal fetuses, which could be explained by the known decrease in left ventricular cardiac output, while increased transit time suggests that there are underlying changes to the microvasculature that facilitate a compensatory increase in oxygen extraction, such as that achieved by vasodilation. Of interest is the greater perfusion deficit seen in the central thalamic region relative to the peripheral brain region. This indicates an attempt to preserve perfusion to the frontotemporal cortex and may represent a compensatory developmental response in late gestation. Subset analysis showed that the global perfusion deficit correlates directly with the degree of pulmonary hypoplasia as measured by normalized lung weights, which suggests that the severity of CDH may influence brain perfusion. At this time, work is ongoing to study the impact of different modalities of ECMO support on cerebral perfusion, with preliminary studies suggesting that the dampening of global cerebral perfusion seen upon initiation of conventional VA ECMO support is not observed in animals on pumpless support. Further work will elucidate whether this improved baseline perfusion translates into preserved autoregulation responses in the cerebral vascular bed. I would like to acknowledge the extremely competent support for these experiments, which was provided by the members of my lab, including the research fellows, technicians, and our medical student, and a particular acknowledgement to Dr. Ryan Didier, a pediatric radiologist at Children's Hospital of Philadelphia, who performed all of the contrast-enhanced ultrasound studies. Thank you very much. Once again, it has been my honor to have this work supported by the Grosfeld Award, and I will be very happy to take any questions at this time.
Good evening, everyone. Welcome to the live question and answer section for this Grosfeld Scholar APSAsode. I'm very excited to be here. I'm Dr. Sean Kunisaki from the Johns Hopkins Children's Center. I'm the chair of the research committee for APSA, and I'll let Dr. Lavorne introduce himself. You're on mute, Bo.
Thank you. Yes, Bo Lovern from Nashville. I serve as vice chair with Sean and am honored to help moderate this session this evening. So congratulations to all of the winners. Dr. Tom Tracy has a question for the speakers regarding what factors they think were most important elements in their various environments that contributed to their success. So maybe we'll take these in order, Dr. Spear, Dr. Grant, and then Dr. Partridge.
Sure. Well, thank you again to the APSA Foundation. As previously mentioned by myself and others, this has really been instrumental. And I think in regards to Tom's question, I think forming a mentorship team is probably the most important thing that I've found that has helped my research progress, because not any one mentor can give you everything that you need. And so I've formed a team with people that can help me get grant funding, with people that can help me with a specific experiment or assay, people who do GI research, people who do surgery. And I think having all those different mentors around you, especially at the beginning of your career, really helps you get the advice and guidance that you wouldn't otherwise have as a young investigator.
Thanks for the question, Dr. Tracy, and I'll echo what Allison said. Thank you so much to the APSA Foundation for the support. And I actually have the same exact answer. When I left fellowship, I was encouraged to start a launch team, which is a group of mentors at my institution as well as outside. And that included my lab PI, my division chief, and several mentors. And that was critical in getting me my protected time and making sure that everyone, including myself, remained accountable. The second most important thing was identifying a great lab PI with a science background to keep me on track and to give me the mentorship and the grant writing support that I needed. And then finally, just all my mentors and partners as well for the support day to day.
And I hate to be a broken record, but a lot of the same sentiments. Awards like this in the early stages of investigation, when you're building your platform and don't necessarily have the breadth of data to support early grant applications, these are really the that allow you to plant seeds of ideas and sort of fund projects that might be a little more risky, high risk, high reward. So awards like this are a great component. In addition to having, mentorship is such an important aspect of scientific growth and development. The other thing I would point to is having the right infrastructure in place. I feel very lucky that, at CHOP, there's a very well-established Center for Fetal Research that was founded by surgeons and is driven by surgeons. We start and end each day in the lab. Our offices are over in the research wing, not in the hospital. So even if we only spend 5 minutes at the beginning and the end of the day in the lab environments and have busy days in the hospital, that touchstone and that sort of infrastructure that exists here, I think was really helpful in setting the groundwork for a lot of what we've done in the lab.
That's fantastic. I also want to maybe, this is a good segue to maybe recognize the contributions of Tracy Grickscheit, who actually was one of my senior residents back in the day at Mass General, who obviously was an incredible mentor to both Dr. Spear and Dr. Grant, who were both research residents in the laboratory. And I suspect that she has a presence in your academic careers even today. Obviously, there were pretty positive experiences. I think that your time was affectionately known in the lab as the G unit, I believe. And so I think it was an inspiration, certainly in your pursuit of pediatric surgery. So I just want to recognize really that there's just an amazing sort of chain of researchers who are really, I think, inspiring this current group of young surgeon scientists. Also want to recognize Dr. Grant. So she, I didn't realize this, but Dr. Grant is the first Black woman, as well as the first woman from an underrepresented minority background to receive the Grosfeld Scholarship in the 25-year history of the APSA Foundation. So I think that's pretty incredible. And so I just wanted to recognize that. And so congratulations to you, Dr. Grant. So are there any other questions in the chat? If not, Dr. Lavorne and I can certainly ask you guys a few questions in about 10 minutes that we have.
Yeah, I see one from Dr. Fallot. And again, I think maybe this time we'll go in reverse order. So we'll have Dr. Partridge speak first, then Dr. Grant, and then Dr. Spear. So Dr. Fallot asks, how do each of you manage to protect your time for research? And how much of your time do you spend doing research?
That's a great question. And I'll quote my chief of surgery, Scott Adzik, who says, on paper, my contract would say that I spend 50% of my time on research and 50% of my time clinical, but in practice, he expects 100 and 100. And I think that really is more accurate. every single day, I'm kind of running back and forth, and the scrub nurses laugh themselves silly when we get calls from the LAM room, with ECMO crises in the middle of cases. But it really is kind of a life of running back and forth between the two worlds. And I really wouldn't have it any other way. Operating on the sheep is obviously less stressful. So it's kind of a nice break and relief. But I do think that, to carve out a bit of time for research in a busy clinical practice, it has to be your hobby. It has to be the thing that you like to do, at 10 o'clock at night, reading papers, whenever you can carve out a little window of time, because your patients will always come first. Your clinical practice will always come first. But if you're in the right place with the right infrastructure and support, I think it certainly can be done. And even if it's very busy, it's just so very rewarding.
So I have 30% protected time, which is about 6 days a month. Initially, when I started, I went every Monday and then some scattered days. But as part of my launch team, Dr. Hayes actually suggested back-to-back days. And so I started doing every Monday, Tuesday, 3 weeks a month when I'm not rounder. That works out okay because my PI's lab meetings are on Monday morning. But I do find myself going to lab multiple other days and working nights and weekends sometimes for the grant writing and doing sort of other tasks. So it is a job that sort of bleeds into the clinical work and you sort of have to carve out the time to do it. So I would say 30% is definitely an on paper estimate. And some weeks I don't see the lab at all because my clinical duties demand that. But overall, I try to spend at least that much time. And I also have a lot of support from my PI's group who's been helpful and started to learn the surgical technique as well. So being able to delegate some of those tasks has helped me get more space for the thinking and the writing.
Yeah, now that I'm last, I'm going to echo a lot of what Emily and Krista said. But for me, it's really it's a top-down protection of time. So my chairman, Kevin Lally, it's his mission of our department for everyone to be 50-50. And as Emily said, that's more on paper because in theory, we're really 100 and 100. But because that's his mission for our department, everyone in the department also believes in that mission. And even just this week, I wanted to do a case that I thought was interesting, that I had seen the patient last week while I was on call. And my chairman reminded me that I should be writing today. And so he did the case, and I've been working on my resubmission of one of my grants. So I think it just takes a team, all of your partners and your division chief and your chairman protecting your time for you and holding you accountable and yourself holding yourself accountable.
That's terrific. I do have kind of a scientific question for each of you, but I'll start with the good Dr. Spear. Since you encountered some unexpected findings and things that may not have fit your initial hypotheses, tell me what other functional readouts would support the enteric nerve system incorporating into these human intestinal organoids. So are there other mechanisms or a functional readout like peristalsis or other factor, or is there a molecular, how does the enteric nervous system interact with these tight junctions, and what could you do at the immunohistochemical level or some other to say they're actually contacting what they need to with these cells? So as you troubleshoot your system and some unexpected results, what other functional readouts are there to verify validity of the ENS cells working?
Thank you for your question, and it's an insightful one. So I have three answers for all of that. Other readouts that we're interested in looking at besides barrier function definitely are peristalsis and motility. So we're actually using a contractility apparatus. Mike Helmrath's group has published that the ENS does improve contractility in an apparatus like that, but we're actually starting to use some agonists and antagonists of the serotonin system to see if we could even improve that further. There is a molecular imaging center here at UT Health, and Eva Sevic is the head of the center, and she does a lot of lymph imaging, and she's published in mice a way to provide quantifiable data on peristalsis using ICG, which is secreted through the biliary system. And so we're hoping that if we can create an anastomosis with our organoids in vivo in mice, we could use that system to provide an in vivo peristalsis readout. You asked about genetics and what role does that play. We're planning on using some knockout pluripotent stem cells for serotonin 4 receptors, which have been shown to influence both contractility and barrier function. And so if we make our neurons or organoids or both or vice versa with those knockouts and without the knockouts, we'll be able to sort of tease out a molecular mechanism for both of those functions. So I think in regards to this specific study done for APSA, when I discussed it with my mentoring committee, Mike Helmrath brought up a good point. My area looked at in our Essing chambers is a lot bigger than other people have used with organoids. And so I think there's some heterogeneity there. And so we're now going back and looking at every single organoid that we put in the Essing chamber and trying to create a grading scale for the ENS that was or wasn't present. And I think if we stratify it by grade of ENS, we actually will see a difference. Thanks again.
Terrific. Dr. Grant, if I could ask you a question, I'm a brother in the oncology world with you. I think your work is fascinating and certainly remember reviewing your proposal with excitement and enthusiasm to see your work come to fruition. One question I have for you is whether upregulating contactin-1 in vitro, did you conduct any colony forming assays or soft agar assays to see what effect that would have on both your NMIC negative and amplified lines? And then what is it about the difference? You showed some paradoxical results that I didn't really expect in differences of functional behavior in these MYC amplified lines. So what I think we know a lot about MYC-in and PI3K and AKT signaling, et cetera, but we don't know a lot in the non-MYC cell line. So tell me a little bit about what differences there are in the signaling pathways going back and forth between the two cell lines. Thank you and congratulations.
Absolutely. Thank you for that. So we actually have not done any ectopic overexpression of contactin-1 yet. That's one of our natural next steps because we have found that contactin-1 is protective overall in neuroblastoma. But we were also surprised to find the differential effect, but it was absolutely always with the MYC-in. MYC-in amplified and MYC-in non-amplified, both with our in vitro and our in vivo studies, everything lined up essentially opposite for the MYC-in amplified and non-amplified. And we can't really explain that just yet. The first experiment we did to try to get at the mechanism was this RNA-seq analysis that I mentioned, and obviously you get a lot of big data readouts for that. But the PI3 kinase pathway came up in essentially every one of the tumors and every group that we compared. Interestingly, contactin-1 has always been found to be a detrimental factor in the literature. So it's been looked at in many adult tumors, never in a pediatric tumor before, and it's always detrimental. So metastasis, lymph node mets, worse invasion of the tumor, and so on. So this is completely opposite of that, and that's what we found most interesting. So how could we exploit this to be protective in neuroblastoma? Is there a way that we can overexpress it with some small molecule mimic? Or is there a way that we can try to block it in the MYC-in amplified tumors? Those are the next steps, essentially. But in terms of mechanism that you were asking about, we're really just at the tip of the iceberg.
Now, I am interested for Dr. Partridge about the pumpless setup, and this is not about cerebral perfusion, which I know was the focus of your study, but more about the model. And so how does the pumpless system work if we have any really exacerbated pulmonary hypertension and cardiac insufficiency or compromise? How is the newborn heart able to pump through that system?
Yeah, it's a great question. So the way that we do it in the lambs is because they are so very unstable and difficult to ventilate, we do it like an exit procedure. So we keep them on placental support while we cannulate. And once we establish circuit flow, then we cut the cord and the animal is essentially delivered. possibly partly because we deliver them so smoothly in that way, we really don't have a lot of issues with cardiac dysfunction, although we do have very severe measures of pulmonary hypertension in the lambs. And, you know, it's very interesting whether you believe the return of oxygenated blood to the pulmonary vascular bed, because the mechanics of this are much more similar to VV ECMO, which is, you know, used in a number of CDH centers preferentially. And, you know, so with carotid outflow and jugular inflow or return of oxygenated blood, are we supersaturating the pulmonary venous bed and inducing some vasodilatation and relaxation as a result? I don't know. We've never had to use any kind of pressor support. At the same time, when we talk about extrapolating this into a clinical pathway, we certainly recognize that some of our patients will already be on some assistive medications like dopamine before they even qualify for ECMO. So we presume that we would, you know, not necessarily expect to wean those rapidly, but that essentially if they have a measurable blood pressure, they'll perfuse the circuit and they'll oxygenate through it. And then it's just a question, I think, of how much additional cardiac support they might need if they do have a lot of dysfunction. We are planning to embark on a clinical trial at CHOP. It's going to be undertaken very carefully. And as I'm sure you can imagine, for the first few babies, we're going to have a prime conventional ECMO circuit right at the bedside, ready to go if there's any sign that they don't like the pumpless system. But so far from everything we've seen in the lambs, it seems to actually treat the pulmonary hypertension. And by the end of the 3 weeks, their echoes actually look pretty healthy and normal.
That's fantastic. Again, I think we're out of time, but I wanted to congratulate Dr. Spear, Dr. Partridge, and Dr. Grant for really an amazing session. I learned an incredible amount about your specific research space. You guys are an inspiration to us all. You guys are amazing surgeons and really amazing role models as well, with an incredibly bright future ahead of you. Congratulations.